good evening. Great to have all of you here. I'm just gonna share my screen now so that we can get started on this. Okay, still people are joining. All right, I'm just gonna share my screen. Can you guys see my screen? Uh, can you see this, for instance?
Yes, ma'am.
Okay, great. All right.
Yes, ma'am.
Okay. So I think whoever is going to join has joined by now. Oh, more people are coming. All right. Okay. All right. We can get started anyhow. As people will come, I will let them in. Okay. So you guys must have familiarized yourselves with the content that was uploaded for week one, right? And so this is going to be a problem solving session that where we tackle some. Okay, one second. Uh, just a sec. Uh, okay, I think that should be fine. Um, hmm. Right. So, sorry about that. Yeah, that we're gonna just go through these questions and maybe look at a few key concepts that were discussed as a part of the week of this week's coursework from your professor. And if you have questions, please feel free to just, unmute yourself and go ahead and ask me the questions and we can discuss. Okay. So let's get started. So the first thing that you have seen in this introduction to cell biology, so the first part of your coursework is, of course, the very important discovery of the cell, right? So Robert Hooke, a very, very curious person, he was very, very excited to study about these different things, and he had very basic stuff available to him. So he's the one who first visualized cells in a section of cork from plants. And he had, as you can see here, an extremely basic setup for himself. He has this really basic microscope. Um, just a second. I have to keep admitting people, which is very annoying. Okay. Yeah. And this very simple microscope that he has over here. In this, you can see this barrel. This barrel contains a bunch of different lenses that can help you focus light, an eyepiece through which the light will finally reach your eye. Over here, you keep your sample or your specimen. And this setup is basically for allowing some light to diffuse onto your sample, and then the sample will reflect this light through this objective, which is used for collecting your light. This focusing screw can help you adjust the focal length between your objective lenses and your specimen, and then this light is all nicely focused into the eyepiece from where you can see it. So this, just using this very simple setup, he was able to make so many extensive notes in his book Micrographia. Now you have to remember that in this time there was no camera, there was nothing that you could just digitally record stuff with. So if you see something, you have to draw it. So that's what he did. Everything he saw with his microscope, he drew, from the very famous sections of cork to insects, plants, whatever. So if you guys want to check it out, it's a very interesting resource where you can look through pages of his notes in Micrographia, right? So I just thought that would be interesting for all of you to uh, take a look at. I just want to quickly recon before I go on to the questions. I hope everyone can see and hear me. This is happening, right? You can see the slides and you can hear my voice.
Yes, ma'am.
Okay, fantastic. All right. So now let's go to some questions. So to those who arrived a little bit later, we discussed that how we're going to do this is I'm going to go through questions one at a time, and you guys think of the answer and just unmute and call out the answer, what you think it is. It's okay if it's wrong, it's not a problem. We'll discuss what the correct answer is and why. Okay, so let's start with the first question. Oh, here. So, which scientist first observed living cells? Do you guys know?
C.
That's correct. So, some might be, thinking that it's Robert Hooke, but he looked at it in cork, and that's actually from dead, those actually dead cells. So, Anton van Leeuwenhoek was the guy who first observed living cells and called them tiny animalcules. Oh, okay. I have to admit someone else. Just a second. Uh, Please just mute yourself. That would be great. I think I can mute this person too. After you're done responding, please just mute yourself. That would make things easier to follow. Just a second. Okay. All right. Okay. So the next question is, which phrase was added later to the cell theory to correct germ theory? So, germ theory, you might remember, was this strange thing that people thought initially that living matter came from non-living matter. So, which of these phrases is the correct one?
A.
That's correct. So, that's
Unicellular cellular.
correct. Very good.
A. Answer
So,
is A.
that's the correct one. It means each cell comes from a pre-existing cell, and this was given by Rudolf Karl Virchow, right? So, that's great. All right. So the next question is, which of the following have a cell wall? What do you guys
D.
think? Please repeat.
D.
D.
D. D.
That's right. Great. Others also answer. I'm hearing just one or two voices. Everyone please join in, okay? So that's completely right. It's D, which is both A and B. So fungi have chitin in their cell walls. Bacteria have a lot of peptidoglycans, while a mouse nerve cell is an animal cell, so it doesn't have a cell wall. So great. That's very easy. So, which of the following are photosynthetic?
B.
B.
That's
B. Answer B.
B. Cyanobacteria. It's cyanobacteria.
Yep, that's completely correct. That's cyanobacteria. So these are also known as blue-green algae, and they have chloroplast, which allow them
Answer
to
B,
make
ma'am.
photosynthesis. Yes, correct. Great. All right. So what about this question? Which of the following is a characteristic of prokaryotic cells?
B.
D. Answer B, ma'am.
D.
They are capable of cell division.
Great, great. That's good. So, yes, they are capable of cell division. And why are the others not correct? Because, of course, they can respire. They have all of that machinery. They have their genetic material. They don't have that bound within a nucleus. They don't have a nucleus. It's just kind of in the cytoplasm. And they don't possess peroxisomes. They also don't possess other membrane bound organelles like mitochondria and the Golgi and so on and so forth. So now the next question is, which of the following is not a characteristic of eukaryotic cells? What do you guys think?
A. They possess a nucleus.
It's not a characteristic of eukaryotic cells. Do they possess a nucleus?
B.
Answer C, ma'am.
B.
That's correct.
C. C.
So some people
Answer
are saying different answers, but someone said the right one, and it's C,
C is answer.
right? Because they have linear DNA that's then bundled up into chromosomes. But in prokaryotes, they have circular DNA, right? So you have heard in bacteria covalently linked circular DNA. So that's what they have. Whereas eukaryotic cells, they have a nucleus. Um, they are not actually uh, necessarily always multicellular. Actually, wait, hold on a second. It's not a characteristic is my bad. Sorry. The right answer is B, guys.
Yes,
Uh,
ma'am. वही तो मैं पूछ रहा
I'm
हूँ.
so sorry. I thought I had written no, they are not always multicellular. My bad. They are not always multicellular. They can be single celled as well. We know many single cell, like the amoeba is a eukaryotic cell that is single cellular. But they have linear DNA that's organized into chromosome. That's absolutely correct. That is a characteristic, and they possess membrane-bound organelles, like I had mentioned earlier. So yeah, sorry, I got distracted. Right. So now the last question of this slide. So cyanobacteria are said to be the bacterial ancestor of which organelle?
Chloroplast.
Yeah, that's right. Very good.
C option, ma'am.
Yeah. Great.
C,
And
C.
this makes complete sense, right? Because cyanobacteria, they are capable of photosynthesis, and chloroplast is an organelle that's found in much more evolved cells that allows them to do that. So it makes sense that maybe, a long time back, these bacteria became sort of endosymbionts that then started living comfortably inside these more developed cells and became a natural part of them, becoming chloroplast. So you've also read and seen in your slides, in your coursework, that mitochondria also were at some point bacteria. And there's a lot of evidence to support that as well, which is very interesting. I'll just let a few more people in, I think. Yeah. All right. Cool. So now we have we have discussed all of these questions. So we'll move on to the next set. So which of these are a typical model organism in research?
D.
Mus
All of
musculus.
all of them.
All of the above.
D answer.
Because ma'am, mus
Option
musculus.
E, ma'am.
Ma'am, mus musculus
All
is
of
a
the
rat,
above.
and Drosophila and Longister is also used in organism. So all of the above.
That's
All of the above,
correct.
ma'am.
So that's right. So after, it's a little confusing because I've put the scientific name. So mus musculus is mouse. Okay. Rat is ratus ratus. Saccharomyces cerevisiae is yeast. Arabidopsis thaliana is the very well known, completely sequenced plant that's used in a lot of plant related research. Arabidopsis. Drosophila is, of course, the fly. So the answer is all of the above. These are all very commonly known um, model organisms that are used in different types of research. So now we come to the region of, different types of bonds that are important in our cells. So how is a covalent bond formed? Do you guys know which one is the answer?
D. D, ma'am. By sharing of pair of electrons.
Ma'am, D.
D or B?
Ma'am, D.
D
D, ma'am.
answer,
D.
ma'am.
Yeah.
B.
D.
Whoever is saying D is a little bit incorrect here, because
A answer.
electrostatic interactions are not necessarily, they are not a part of covalent. There are non-covalent interactions, electrostatic interactions. So
Okay,
that's
ma'am.
not right. Uh, C is also not correct because it's not a complete donation from one element to another element. So it's B, which is sharing of a pair of electrons between two elements. Now, why is A not correct? Because, yeah, I mean, they're sharing, but is it always equal sharing? So you guys might have seen in your coursework that there was something called a polar covalent bond. So that is when one say atom is more electronegative than the other, so it is pulling that electron pair a little closer to itself. So it's becoming a little bit negative and the other is becoming a little bit positive. So it's a slightly unequal sharing, but it's not a complete donation of bond either, of the electron either. Okay. So the answer here is B, right? Sharing of a pair of electrons either equally or unequally. All right. What about this question? So amino acids are covalently linked by what kind of bonds? There's a name for them. Do you guys know?
Peptide bond.
That's correct.
Covalent bonds.
Yeah, it is a covalent bond. It's a and the name, like the type of covalent, it's like the covalent bond is a type, right? So this is formed by a peptide bond. Someone said it earlier. So it's formed by the exclusion of a water molecule when your COH and NH2 of neighboring amino acids will react. So that forms this very nice, strong covalent link called the peptide bond. Okay. And the next thing is secondary and tertiary structure of the
Hydrogen bond.
involves, yeah, that's correct. Hydrogen
Hydrogen
bonds.
bond, ma'am.
There's some other stuff as well. Do you guys know the other kinds of interactions that might be involved?
Wonderful forces.
Yeah, yeah.
Hydrogen bond, ma'am.
Yeah, hydrogen bond is one. Van der Waals forces, definitely. There's one more, actually two more that are there. Does anyone know? While you guys think, I will just admit this one person. Okay. Okay. So, some other forces are these disulfide bridges. So this is something you see interestingly. Have you guys ever straightened your hair? So hair has these disulfide bridge secondary structures that you can break when you heat your hair, and that's why it becomes straight. But the curled part of the hair has these disulfide bridges, which is interesting, right? And there's also these hydrophobic interactions that come from basically the exclusion of amino acids that have hydrophobic regions from water loving areas. So these are the different kinds of interactions, around 3, 4 of them, that are important for secondary and tertiary structures of proteins when they're folding into sort of more complex structures, right? So the primary disulfide linkages, the peptide bonds are what are necessary for forming your polypeptide chains of amino acids. And then when you want to fold it into something more complicated that's actually functional, that time you have all of these other different forces that come in, right? So now, what about this one? Which of the following is not a characteristic of hydrogen bonds? Do you guys know?
Ma'am, B option.
Answer B.
Yes?
B.
Great.
B, B.
Good job. All of you have got it. It's straightforward. So they're weaker than covalent bonds, about 1/20th weaker than covalent bonds, but still they're so important in holding our entire body together, really. And they are only seen between water molecules. This is not true. You can see it even between, say, if you replace the H2O ka O with F or something, even then you can see hydrogen bonds. It all depends on the electronegativity of that group, and we know that these fluoride, chloride, all of these are very strongly electronegative, so they like to draw this electron cloud towards themselves. Yeah. And so that's great. So now we have these many questions solved. What about this one? Which of the following is not true about carbon in the body? This is easy. Let's see.
B, ma'am.
See.
C.
Right, perfect. Great. So that's easy. Of course, it forms stable bonds with other carbon atoms, right? That's just a basis of so many different structures. And of course, it can form covalent bonds because it has four empty spaces for it to have electron in. And it also is found in combination with H, O, and N, and also S. There are some sulfur containing proteins as well, which I haven't mentioned here. And of course, it's present in human muscles because muscles, after all, are made from a bunch of different proteins, and proteins have carbon. So that's great. Before I move on, I think there's someone else trying to join. Great. Okay. Oh, there's one more question. Right. So this is very easy question, but what is the enzyme called that's required for ATP hydrolysis?
ATPase.
Yeah, you got it. So DNAs, RNAs, ATP is here. So now we'll move on to actually looking at these cells. So you guys learned about animal cells and plant cells. And you know the differences between them. So you know that in animal cells, You have these different membrane-bound organelles, such as the mitochondrion, the centriole, very important for cell division that you'll learn about in more detail later. Peroxisomes, where a lot of these oxidative interactions take place. When the cell is stressed, peroxisomes are very important for trying to reduce the amount of reactive oxygen species that are roaming around in the cell and damaging stuff. You have your rough endoplasmic reticulum that have your these lots of ribosomes. Um, Smooth endoplasmic reticulum, the lysosome, which as you all must have heard in school and stuff, they're called the suicide bags. So they're very important. They have a lot of enzymes in them that can destroy uh, anything really in the cell that needs to be destroyed. There are some proteins that have been folded badly. They can be targeted and taken into the lysosome. Something that has entered the cell that shouldn't be there can be destroyed by the lysosome. Then you have, of course, your Golgi apparatus, important for sorting and transport through from the cis side to the trans side. Then you have the nucleolus within the nucleus, and of course, your nucleus in the animal cell. And very importantly, the cytoskeleton, which includes your microtubules, actin, and intermediate filaments. And these are very, very important for not just maintaining the shape of the cell, but also how the cell is able to interact with other cells, how it's able to move, how it's able to divide. All of this is facilitated by the cytoskeleton. So it's an extremely, extremely important part of the cell. Similarly, also in plants, you have a much larger vacuole, turns out, and you also have your mitochondria, your cytoskeleton, peroxisomes. Of course, the major added thing are two things. One is the chloroplast, which is important for photosynthesis, and the cell wall, which is giving it a much, much more rigid structure than your animal cells. So with this in mind, I thought I'd show you guys this. These are TEM images. So TEM is transmission electron microscopy. So this is a slightly modified version, volume electron microscopy, where you can get a little more understanding of a thicker sample. And this is really, how these structures look. So you can see in this one, they have seen the plasma membrane over here. This is the extracellular space. Here's the cytosol. These are mitochondria that are seen with transmission electron microscopy. An endosome, the ER that you can see, these slivers of. This is the mitochondrial membrane, mitochondrial ribosomes. And here also you have, I'd seen a lysosome somewhere. Yeah, you have lysosomes over here, these black bags. So when something looks dark in these kind of images, it means they're electron dense. So these clearly have a lot of stuff packed into them, very densely packed, electron dense. So you can see this lysosome very clearly over here. Here you can see the Golgi. It looks so much like in your textbooks, right? And these different lumens of these vesicles, these little vesicles that come in and out of the Golgi. Then this is the nucleus. So the membrane of the nucleus, this NP in and NP out, these are nuclear pores. So this is what helps facilitate stuff going into the nucleus and coming out of the nucleus. Then you have your nucleus in more detail. You have the nucleolus, nucleoplasm, right? So I thought that this might be interesting. And of course, yeah, the centrosomes and their very famous sort of T or H-like structure. So this might be interesting because we see it as diagrams, but where do those diagrams come from, right? It comes from these kind of very high resolution, painstaking imaging studies that have allowed us to see these organelles at in such detail, right? So these are HeLa cells. So HeLa cells are basically they're human origin cells. They're a cell line, tumor cell line. So this is a eukaryotic animal cell, you can say. And if you want to look at more of these images, this paper and some others that I'll attach in the references when I've shared these slides, you can check them out. They're super cool. Okay. I think someone was trying to enter. Let me just see. Oh, okay. It's just a hand. Someone has a question. Ankita, do you have a question? No? Not anymore, I guess. Okay. All right. So I'll move to the next couple of questions. So what is the primary function of the nucleolus in the eukaryotic cell? Can you guys tell?
A.
A. Not entirely. Does anyone else have a different answer? So this is the nucleolus, okay, not the nucleus.
B. Ribosomal RNA synthesis.
B.
That's correct. Yeah, it is B. It is ribosomal RNA synthesis. So another tidbit of information is that the nucleolus itself is not membrane bound, which is interesting, right? You'd think, how are all of these proteins just all together inside the nucleus and not just spilling everywhere? Why aren't they just diffusing everywhere? So there's a lot of interesting research, and people try to find out, okay, why is it that the proteins and all of the components of the nucleolus are able to just be in one place without a membrane? And it turns out it's to do with phase transition. So they actually are so concentrated that they come out of solution, and they are sort of like separated out from the solution, which allows them to sort of clump together. So that's another interesting thing. Okay, so the next question. Which organelle is responsible for sorting and transporting proteins destined for secretion?
Golgi apparatus. Golgi apparatus, ma'am.
That's correct. Yeah, it's not the endoplasmic reticulum, as I think someone was saying. It's the Golgi. Golgi is very important for taking inputs from the ER. So ER has a lot of these protein processing functions, and then the proteins move from there to the Golgi and are and move through the Golgi, undergo several different post-translational modifications there as well, and come out from the other end. And then they can either be secreted or they can go and interact with something, whatever, based on what the cell needs. Okay. So what type of ER, endoplasmic reticulum, is associated with ribosomes?
B.
B.
B, ma'am. B. Rough endoplasmic.
Yep, that's correct. Really good. So that's an easy one. All right, so let's see what the next question is. So this is a little bit of reading here. Which of the following is false about the plasma membrane in animal cells? So what do you guys think? Read carefully and tell me.
B, I think.
Anyone else? Any other answers that others have?
A.
C.
Okay, so basically people have all answered different stuff. So this one clearly is a little confusing. So I'll tell you guys the answer. One person said it was C. Okay, so I'll explain why. So you know that in the plasma membrane, right, it's a lipid bilayer. That means there's an outer layer or a leaflet and an inner layer or leaflet. And the outer and inner layer have different compositions of lipids and proteins. So it has been taught also in your coursework that there is asymmetry of lipids in these in the plasma membrane. So whatever number of whatever the composition is of the outer leaflet may not be the same as the inner leaflet. For instance, you might have, like in that's let's said in the B point, which is cholesterol. It's actually found in higher amounts on the outer leaflet than the inner leaflet. But this is not necessarily this is not a bad thing. In fact, it's very important. It's important for signaling, it's important for cell structure, it's important for the ability of the different proteins to move around in the membrane. So this composition is actually very important for homeostatic condition. That means normal conditions of the cell. For day to day, functions of the cell, you need this asymmetry of lipids. Whereas when it comes to point A, you're seeing there's lateral diffusivity, rotation, flexion, flip-flopping of lipids. So this is actually totally normal. These conditions, these different phenomena do occur where, as you had seen in the modified Singer-Nicholson model of the plasma membrane, you have these lipids that do diffuse around, and this does allow a lot of dynamic moving of these proteins and lipids in the membrane. Rotation of the lipids does definitely occurs with the help of these fatty acid tails, flexion, flip-flopping of the lipids. This is all normal and this all occurs. So the only thing that was false is C. And D is true because plasma membranes can either span across the membrane proteins can either span across the plasma membrane. These are your transmembrane proteins. You have a lot of these, pore forming, these pore proteins, ion channels, and these different types of transmembrane proteins. Or you can have these signaling proteins involved in just being anchored to one side, like GPI anchored proteins. So these also exist. So that's why C is the false one. Okay. So now this is one interesting example actually I thought I would give with respect to why asymmetry is important and how the changing asymmetry can actually be a non like a, be something that you see in a non-homeostatic condition. So here you'll see that there's a healthy cell, okay, that has some sort of, it has a normal sort of round look. And then when it's dying, it starts forming these blebs and it gets really weird. And this is called apoptosis. So the regulated death of a cell is called apoptosis. Now you'll see below the healthy cell in that diagram of the lipid membrane, you can see that phosphatidylserine, which is a type of it's a type of a phospholipid that's present in the plasma membrane. It's more on the inner side of the membrane, right? But during apoptosis, this PS or phosphatidylserine comes onto the outer membrane. And this is not normal, right? The cell, when it's dying, it's very unhealthy. So the body needs to know that, okay, there is a dying cell over here. And how does it know? One of the ways it knows is that this PS comes onto the outer leaflet, and that is acts like a signal. So then other cells that are used for clearing up dead cells so that they're not, harming other cells, they will come, these clearing up cells like macrophages and so on, and they can detect this PS signal and take up this dying cell. So here the asymmetry was what was normal, and then when we reversed that and brought all the PS onto the outside, then the cell was undergoing apoptosis and it acted as a signal for a dying cell. So that's one example that I thought you guys might find interesting. Right. So let's move on to some more questions. I will just check before that if anyone is left to join. Yeah, there are. Okay. Okay. I think everyone's here now. Okay. So we'll move on to the next question, question number 18. So as you know, there are transmembrane proteins that extend across the plasma membrane, right? So for this to be possible, the composition of the transmembrane region of these proteins is actually maybe a little different from the rest of the protein. So it can be termed as either polar or non-polar. So what do you guys think? And I'll explain why based on your answer. Is the transmembrane domain of proteins that span across the membrane, is the region that is embedded within the lipid bilayer, is it polar?
Ma'am, non-polar.
Yeah,
Ma'am, non-polar. Because the membrane is also non-polar. I mean,
exactly.
lipid.
That's right. Exactly. So there's the fatty acids, right? That the fatty acid tails of these lipids that are all pointed inwards in the lipid bilayer, and those are obviously highly hydrophobic and highly non-polar. So if you want a protein to be able to go across a membrane, you need that transmembrane domain to be non-polar so that it can stably be inside the membrane. Okay, so great, good. And guys, please let me know if you have any questions or any confusions. Okay, so ion transporters transport ions in a certain way across the membrane. So do they use energy or not? Based on that, it can either be passive or active. So do you guys know whether it is passive or active?
Active.
That's correct. Yeah. So they transport ions actively across the membrane. So this is with the help of energy. So they need energy because it might be against. Oh shit! I said the answer because it's against the concentration gradient.
Yes, ma'am.
Yeah. Yeah, so that's what ion transporters do, and you might have also seen that these ion channels sometimes just go with the diffusion gradient. So if you have, say, more chloride ions outside of the cell, then you will move them across the concentration gradient into the cell where there is less chloride ions at that point. So that is a passive transport mechanism with the help of certain ion transporting channels. But these ion transporters, they often have some sort of a ATP pump that allows you to move them across a gradient. So you're actually expending energy to do this. Okay, another question. Complex food molecules are broken down within the cell in what organelle? So this is eukaryotic cells, obviously. Anyone knows? Can you guys hear me? Oh, I hope you guys can still hear me. Okay.
Yes, ma'am.
Okay. Everyone went silent. So, okay. So, if you guys are not sure, so the answer is lysosomes. So, lysosomes are a membrane-bound organelle that I was telling you about a little while earlier.
Ma'am, but
And
it was a suicidal bag. That was I was thinking. That's why I'm not answering. Why is it like broken down? Complex food molecules are broken down within the cell with the
That's
help of
yeah,
lysosome.
that's a good question. So, lysosomes are not just for degrading the cell itself. That is a last resort. If the cell has become old, has become unhealthy, has become infected, or it just has some, yeah, it's a very normal process, then the cell will start eating up the contents and, producing some breakdown products that can help other cells. That is the why the lysosome is called a suicide bag in that case. But normally also in normal conditions when the cell is just respiring and taking in food molecules to produce energy, that time lysosomes also are part of breaking down complex food molecules with the help of dash. So what is the second dash that will help you answer this question that you just asked?
Lysosomal enzyme.
That's correct. Exactly. So lysosomal enzymes are very important for being able to break down complex molecules, including in the process of autophagy, where the cell is breaking down its own components because the cell no longer needs to survive at that point. And one slightly out of coursework question is, what is the pH of this organelle typically?
Below 3.
Yeah. So it's acidic basically. It's a very acidic um, organelle because these enzymes function best at the low pH. And that's actually a really great way people have determined to be able to track these lysosomes. You have different dyes and different sort of reagents that can be used to specifically bind to the lysosome just because it is so acidic in comparison to the rest of the cell. So it really helps with being able to track lysosomes using fluorescence microscopy. And it's something that we do in my lab as well. Okay, here is a respiration question. So what are the end products of glycolysis?
All of the above.
Yeah. Does anyone have any other answers? No, right? Everyone agrees? Great. So that's correct. It is D, all of the above. So you get two pyruvates that come out of glycolysis, and then that undergoes further processing and then eventually is converted to acetyl CoA, which then goes into the Krebs cycle, which then assists with the electron transport chain, and then you get a whole bunch of energy, right? So that was what was shown to you in your coursework, just as a recap. So what is this process of production of acetyl CoA from pyruvate called? You guys remember?
Krebs cycle,
Not
citric
really.
acid cycle.
No. That's one step further. So there's somehow we're forming this acetyl CoA, right? Then acetyl CoA is being fed into the citric acid or Krebs cycle. But how are we getting acetyl CoA?
Link reaction.
Yeah, you have one oxidative decarboxylation, right? So this pyruvate that's coming out of glycolysis with the help of CoA and NAD plus and oxygen you actually convert it to acetyl CoA and you get some CO2 and NADH and all of that. This is
Ma'am,
taking
this is
place.
called as a beta oxidation, I think.
No, no, that is for lipids. That's different. Yeah.
Pyruvate decarboxylation.
Yeah, that's right. So that is how you get acetyl CoA. So I think we've answered that now. So what the next question is about oxidative phosphorylation. So you learned about how after acetyl CoA is produced, it's fed into the Krebs cycle, and then you have these many different enzyme involved steps that eventually throw out one OA and give you a CoA back, and then you have ATP, NADH, FADH2, CO2, and all of this produced. So where is all of this happening, and where are the next steps also? Where are all of them taking place in the cell?
Mitochondria, ma'am.
Yes, where in the mitochondria is it taking place? Is it on the outer membrane, inner membrane, in
Inner
the
membrane.
membrane space?
Is it inner membrane?
Not entirely. So the Krebs cycle is actually taking place in the mitochondrial matrix. It's happening inside the mitochondria completely. Okay. And then the next steps after that, that is when you are moving to the when your electron transport chain is so basically you have your NAD and FADH2 helping with all of this movement of electrons across these different complexes. That is on your inner mitochondrial membrane, but it starts off with Krebs cycle inside the mitochondrial matrix. Okay, so this is how what it looks like to jog all of your memories, right?
Excuse me, ma'am. So, what would be the answer? Is it exactly mitochondrial inner membrane or something else?
I'd say it's probably both. Yeah, both of these things. And yeah, okay. Before I move on, I'll just admit a few more people. Okay, great. So now we have tackled all the way till here. So do you guys have any questions till now, or should I move on?
No, ma'am.
Yeah, it's all good. Great. So, okay. Now we'll talk a little bit more about respiration here. And there was a unit within your coursework about ATP synthase. And it's a really cool part of the respiration process because it actually involves like this machine, basically, which is what ATP synthase is. So ATP synthase is embedded in which portion of the mitochondria? Do you guys know?
It's mitochondrial crystal.
That's correct. So that's basically the inner mitochondrial membrane that is formed into these cristae structures. Very good. So that's where ATP synthase is. And as you know that there is an F1 and F naught region of ATP synthase. So which is the part that's rotating? There was, you might have seen in your coursework that there's a rotating portion. So do you guys remember which one it was? So
Stock subunit.
sorry,
Stock subunit.
the stalk subunit. Not exactly. So there is one ring.
There is also C ring, something type of C ring present.
Yeah. So there's a rotor ring present, which is the F0 subunit. That is within that's within the inner mitochondrial membrane. So it's in the transmembrane region. So I'll show you the diagram in just a minute. It's part of this slide. So there was a head region which was in the matrix, and then your F0 region which is in embedded in the membrane. And that is the rotor ring. That is where this actual machine stuff is happening. Okay, and that F naught region is the rotating portion of ATP synthase. Okay. All right. So now, because I've been saying machine so much, this is going to become very easy for you to answer. But ATP synthase on the whole, it converts what kind of force into what kind of energy? So as you remember, in for ATP synthase, what happens is that you have these um, you have these H plus ions that are moving
Mechanical
through
force to chemical force.
to chemical energy. That's correct. Yeah, that's great. So you got it. So you have these H plus ions that are moving via this concentration gradient that was established with the help of the other components of the electron transport chain. And when these H plus ions move through, the F naught ring is going to start rotating very fast. And this generates mechanical force, and that force is then allows the change in conformation of the F1 region that contains these different catalytic subunits, and that allows the conversion of ADP to ATP, and that is your energy there. So you're ultimately converting mechanical force into chemical energy. Okay, so it's very interesting actually. So now here is a small maths problem, very easy. How many ATP molecules can ATP synthase produce in 1 second? So I don't know if you guys remember the numbers that were given as a part of your video, like what these different parameters were. So I've given a little hint. You need to know the revolutions per minute. So how many times that F naught ring is revolving, that will give you how many times, and then you multiply that with the number of ATP molecules that are typically produced with each revolution. And then you can divide it by 60 seconds. So do you guys remember how many revolutions per minute is this F naught undergoes when you're passing, say, 3-4 ions in one go? Any idea? I think maybe you guys have forgotten, huh? So that's okay. So there is 8,000. Okay, so there's 8,000 revolutions per minute of this F naught ring. So I said now it's fast. So it's rotating really fast. 8,000 rotations per minute. And with every one of them, you produce 3 ATP molecules. So now with this information, can you calculate how many ATP molecules it produces per second?
Basically, it would be 100 to 200 per second.
There's actually
It,
a number. You can do the maths here if you guys have like a pen and pencil. So, I mean, you're close with your guess, but you can be much closer if you calculate. So, revolutions per minute, 8,000, right? Maybe I can write it in the slide for you. So, there's 8,000 revolutions per minute, and the number of ATP molecules is 3. So how would you do this, right? It would be 8,000 multiplied by 3. So that's the total number of revolutions, sorry, total number of ATP molecules across all of the revolutions in 1 minute. So then you just divide it by 60. And so
then
that
it is
is
400.
exactly. So that divided by 60 will be 400, right? So the total number of ATP molecules that are produced per second by an ATP synthase is 400. So think how um, sort of efficient a process this is, that one single ATP synthase molecule can do all of this. But of course, it wouldn't be able to do this if you didn't already have this sort of chemiosmotic gradient set up by all the other components of the electron transport chain. Okay. Great. So that is the end of the first part, which was involving the introduction into the different components of your cell and also a little bit about bioenergetics um, within the cell of within eukaryotic cells in this case. But similar things are seen in prokaryotic cells as well, except they're just not membrane bound. You have all these components distributed along the plasma membrane itself. Um, so now, do you guys want a break before we move on to the next part, or you're fine? We can take a
Break,
break
ma'am.
till 7 and then we can restart at 7. Is that okay?
Yes, ma'am.
Yeah? Okay. All right. So I'm gonna unshare my screen. So you don't need to leave or anything. Just, come back as and when at 7 o'clock, and then we'll restart with the next part. Okay, that's some interesting questions there as well. For those joining now, we just have a short break going on between the two different coursework contents. We'll restart in about maybe 2 minutes or 3 minutes. So just hang on. I'll continue.
Okay, ma'am.
So, hi everyone, to those who were there from before as well as those who have joined. We were just taking a break. Now we're going to go ahead and start with the next portion of the discussion and the different questions that I have ready for you guys. This will be about DNA, the genetic material, that portion of your coursework. So I'll just share my screen now. Okay. So I hope my voice is audible to all of you as well as my screen that I'm sharing. I hope you can see the PPT. Just let me know once.
Yes, ma'am.
Okay,
Yes, ma'am.
great. I'll just admit this person. Okay. All right. So I think we can get started with the next part and move through the questions and wrap it up by 8 o'clock. So, all right. This very interesting experiment must have been, explained in your coursework as well. I thought I'd just go over it because it's something that really was the defining sort of few experiments that started off all of this research into how DNA is very possibly a very important genetic material for cells to be able to pass on information from one cell to its next generation of cells. So the very first experiment was this Griffith's transformation experiment, where you had these different strains of Streptococcus pneumoniae, a rough strain and a smooth strain. So the rough strain was known to be, it's a little blurry, but I hope you can see it. It was a non-virulent strain. So if you inject it into a mouse, the mouse will survive. Smooth strain is a virulent one. So if you inject it into the mouse, that mouse will eventually die. Now, if you heat kill the smooth strain, it's no longer alive, but whether its components are intact or not, that was not something people had thought of at the time. So you inject the heat-killed smooth strain into the mouse will live. But if you mix the rough one and the heat-killed one together, then what happens is somehow the mouse dies. So the question became how come, right? Heat killed smooth strain on its own, mouse is surviving. Rough strain on its own, it's non-virulent, mouse is surviving. Why is it that when you mix these two things, the mouse is dying? So something in this heat killed smooth strain must have got into the rough strain and caused it to change or transform and cause the mouse to die. So something made this non-virulent strain virulent to some degree such that this mouse would die. So how come? So this was Griffith's transformation experiment, and a lot of people got very interested by this. And these three scientists then went ahead and performed a very important experiment where they took these heat-killed S strain cells and they just have like a mix of everything that was inside it. Okay, so lipids, sugars, genetic material, which of course they didn't know too much about genetic material and what it does really. They just knew that there's some component like that, but really what it's doing or how it's going from one cell to another wasn't known. Then what they did is they got rid of each component one by one. So in the first tube, you can see no protein. That means they added a proteinase. That's an enzyme that can just cut up the protein and make it non-functional. So you don't have protein there. Then they added this treated concoction to your R cells. And did they see the S cells appear or not? Yes, they did. So S cells appeared, and when they injected it into the mouse died. Next case, they tried RNAs. So could it be that RNAs is what is transferring this virulence information from one cell to another? So they added RNAs to get rid of RNA and they did the same thing and the same thing happened. So S cells again appeared and the mouse will therefore die. And the last thing they did is got rid of the DNA. So they added DNA as enzyme, and here of course they got only R cells and no S cells appeared. So the mouse would survive, indicating that the information of this virulence was transferred with the help of DNA from this heat-killed smooth strain to the rough strain, making it virulent, therefore. So this was a very important experiment and set up a lot of the basis for why people started looking into genetic material in the first place, why people started looking into DNA in the first place, right? I'll just, yeah, okay. Okay, all right, everyone has a, okay. So now some questions again, let's jump into what you have learned about the structure of DNA, and we'll further move on after that to transcription and translation and all of that. So when you think of the pentose sugar ring of DNA, we know that when you compare it to RNA, it doesn't have one OH group. That's why it's deoxyribose. So on which carbon is this OH group lacking? Which is it not there in DNA and it's there in RNA? Do you guys know? For those who joined late, what we're doing here is we're reading out these questions, and then whoever knows the answer, you can just unmute and call out the answer and then mute again. And if there's any confusions, questions, of course, we can discuss. Okay. So
C2. C2.
anyone else? Everyone agrees? Does anyone have any other answer? Then you're right. It is C2. That is the carbon on which it is. The OH is present in RNA but not in DNA. And I'll show you the structure to jog the memory of anyone who might have forgotten, because these do get confusing and I completely relate to that. So which position of the pentose sugar ring are the phosphate groups and nitrogenous base present on respectively? So do you guys remember that on your sugar ring, you will have your phosphate group that helps linking one base pair to the next, and then you have your nitrogenous base, right, which is your adenine, guanine, all of that. So do you guys know which one is on which carbon of this sugar ring?
C5, C1, it's D.
Any other answers? Anyone else has any other answers?
C4, C5.
What was that?
Ma'am, C4, C5.
Okay. So actually the correct answer is C5 and C1. You can see this diagram here. This will help you remember. So see how C1 over there, that has the nitrogenous base, and C5, which is the carbon that's actually outside of the ring, that is like it's connected to the ring but not directly as part of the component of the ring. That's where the phosphate group is there. So you see, so C5 is the phosphate group and C1 is the nitrogenous base. So it's option D. Okay, D for dog. Right, now here is a math question. So please don't curse me. If adenine, so now before I jump into this question, maybe I'll just give you a background in case you've forgotten. So there's this Chargaff rule that was determined by Erwin Chargaff, that essentially said that the ratio of A to T is similar in the DNA and the ratio of C to T is similar in the DNA, right? And everything will obviously add up to 100%. So now with this information, if adenine or A makes 30% of the DNA, then how much percent will cytosine be? Can you guys
Ma'am, C,
calculate?
20%. C.
Ma'am, I want to ask A and T will be equal or A and C will be equal?
So you know that there is base pairing, right, in DNA. So A
70%.
binds to which one? A binds to
T.
T,
T.
correct. So if you have 30% A and it's binding to T, then there will be 30% T, right? So
Yes, ma'am.
adenine and
Thymine.
exactly, they are 30% each. So if A and T are making up 30 plus 30, which is 60% of the DNA, then naturally C and G will be the rest of it. which is
Together
40%.
40%.
That's
Okay.
correct. And they are also an equal ratio because they are also
20.
bound to each other.
20.
Correct. So the answer becomes 20%. So C and G individually are 20%, and A and T are individually 30%, totally making up to 100%. So this is approximately the case for the DNA. Of course, there are some regions, towards the end and all, you'll have slightly single-stranded regions, not very long. So maybe that will change the number by just a little bit. But overall, this holds true. So this is just for uh, your memory. Okay, so great. So this was a interesting question. Now, okay, why isn't it moving? Okay. All right. So now a little bit sort of out of the box question, but do you guys remember what technique was used by Rosalind Franklin and her colleagues to determine the structure of DNA?
X-ray crystallography.
Yeah,
X-ray diffraction.
yeah, correct. So I thought I'd just tell you a little bit about X-ray diffraction because it's such a complicated technique, and still there aren't a lot of people who know how to do it and solve these structures that you get. And Rosalind Franklin and her colleagues did a really amazing job. So really what happens here, this is a very, very simplified version of this. Um, you have an X-ray tube that's emitting these X-rays. A LED screen, so LED will stop basically sort of these diffused rays from here and there, and you have a little point here that allows what is known as a collimated beam. So all of them are sort of parallel to each other, and so they're very well concentrated onto this sample. So in this case, this was DNA for them. You can use this for proteins as well. And this X-ray goes and hits the atoms within this DNA. It hits the different components, and that, based on where they are positioned, they diffract from here and form this diffraction pattern on a photographic plate. And from this photographic plate, you can then solve, based on known information about how different atoms typically are, how far they are from each other, you can determine an unknown structure. So that's what they did for DNA. And this spot in the middle is just the parallel line. So that's the one that goes right through. So that will be like an empty space. And around it, you will have different diffracted spots from different components of the DNA helix. And that's how they solve this structure of initially just the B form of DNA. And so that was Rosalind Franklin. And this was the famous photo 51 that she had shared from her X-ray diffraction of the DNA helix. So this is what it looked like. And then, you do a lot of maths after this to figure out sort of what distances all of these are from each other and what molecules they are, therefore. So now a question about the DNA double helix. So which of the following is false about the DNA double helix? Can you guys answer?
Ma'am, A.
Option B.
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 Bees.
That's right. I think everyone got it. So that's it's good that all of you know. So we know that strands are anti-parallel. One is running 5' to 3', other is running just opposite to that, 3' to 5'. Then cytosine pairs with guanine, and it is through hydrogen bonds, but it's through 3 hydrogen bonds. And A and T will, as you know, pair with 2. And that tidbit of information for you here is that, say you want to separate the strands of DNA. Okay, so you have your double helix and you want to separate them and get a single strand. So we do this a lot in techniques like PCR. You must have heard of polymerase chain reaction, where you want to separate a double helix. You want to separate them into 2 strands and get one strand on its own, so you can form many copies using it, right? So for that, what you need to do is actually heat the DNA. If you heat it, it'll actually denature it. You'll be able to take it apart into 2 strands. And when we're heating, we actually have to take into consideration how many C's and G's are there in that DNA. So if I have some DNA of some X number of base pairs, I should actually know the sequence and be able to figure out how many C's and G's are there I can count it or use some software to do it for me, and that will help me determine how much I should heat it. Because essentially, if you have more triple hydrogen bonds, it's going to be harder to break the strands apart, right? It's going to be harder to take them apart, so you have to heat it more. So more CG pairs, more you have to heat. And then AT is typically regions that you can separate out easily. So that's something you take into consideration, right? When you're doing these different DNA denaturing steps with heat. Then, of course, one full turn is 3.4 nanometers or 34 angstrom. And the radius of the helix is 10 angstrom. So the total diameter, therefore, becomes 20 angstrom. So great, that is solved. Okay, so here's a question. RNA transfers information about the genetic sequence from what part of the cell to what part of the cell?
Nucleus to cytoplasm.
Okay, great. That one was very easy. So we know that DNA is there inside the nucleus. It's getting replicated there. But your translation and all of that is taking place in the cytoplasm. All that machinery, proteins, all of that stuff is there in the cytoplasm. So someone is coming out with DNA related information to the cytoplasm. And one bonus question is from your coursework, if you remember. Do you guys remember what was one of the diseases, genetic diseases, that led to people questioning whether there is an RNA that's or some intermediate that helps transfer information? There's one blood related disease.
Sickle cell anemia.
Correct. Very good. So people thought that, there is this change in the amino acid, right? There is one amino acid being substituted for another, and that protein is there in the cytoplasm. So how is it that something that's changed in the DNA is also reflecting in the cytoplasm if the two things are in two different regions of the cell? So there has to be an intermediate, something that is taking information from one region to another. So that's when this whole question of RNA came. And people found out more about the dogma of life, central dogma of life. So now one easy question. 3 RNA bases will make one what that makes one amino acid. So there is a word for this.
Codon.
Correct. Very good. So others also answer, huh? So that's correct. 3 RNA bases. When you have the complementary RNA bases that are there in your tRNA, that allows for a single amino acid to be transferred to the growing polypeptide chain during translation. Just
Quiz is going.
at this point, who is joining? I think everyone is there. All right. Okay. So now, reverse transcriptase. This, it was discovered as a very important part and a modification to the central dogma. This synthesizes what molecule from what molecule?
DNA from RNA.
DNA from RNA.
Correct. So it's basically going opposite to what people had thought was the only way it goes the entire time. And this is seen in a lot. This is seen in a lot of these viruses that replicate, right? They have RNA-based genomes, and this reverse transcriptase is an important protein that's found within the virus. So relating to that, these viruses, what kind of viruses are they? There's a name for them.
Reverse transcriptase.
Sorry.
Retroviruses. Retrovirus.
Correct. Very good. So retroviruses, COVID, yeah, the COVID virus, HIV, RSV, many others. These are very well known and dangerous retroviruses. The synthesis of RNA proceeds in which direction? From what side of the DNA to what side of the DNA? So transcription basically, or synthesis of RNA, it proceeds in which direction?
3 5 dash.
Fastest redirection.
5 to 3 direction. That's correct. So from the 5' end to the 3' end, in that direction, that's how transcription takes place. Right. So that's another question. So phosphorylation of what region of this enzyme that's important, this protein that's important for transcription allows promoter escape and elongation to occur.
CTD of the RNA polymer.
That's exactly correct. That's very good. So, do you know why we need to phosphorylate the carboxy terminal domain of the RNA pol? So there are, there's two reasons for that. One is promoter escape. So we know that RNA pol actually needs to bind via its CTD to the promoter, enhancer, all of that complex that's there. And when it binds, it has to be in the unphosphorylated form. So by phosphorylating it, essentially you're allowing it, once it has already bound, to then escape from that and continue with elongating the mRNA. And the other is for the help of these elongation factors. Actually, they prefer to bind to the phosphorylated form of RNA pol. So these two things will happen once you have phosphorylated RNA pol. Okay. So this is the eukaryotic transcription process. This is just a very simplified form of that, and this is what I was saying, that this RNA pol 2 comes, binds to the promoter, and then you have this mRNA that starts forming. However, it's only able to move away from the promoter and keep going along and elongating this mRNA, keep moving along the DNA and reading more of it, only once it has been phosphorylated in the CTD. And then it reaches a termination site, and that's when the synthesized mRNA can be ejected, and this RNA pol also comes off. So that's just a very simplified form of it. I'm sure you guys will read more about it in like upcoming course stuff. So next question, what is the enzyme that's required for charging the tRNA? This is during translation.
RNA polymerase 3.
Does anyone have any other answers?
Amino acyl tRNA synthetase.
Yeah, that's right. Yeah, so that's correct. It's amino acyl tRNA synthetase that's important for charging tRNA and extending the polypeptide chain during translation. Next is an easy question. So, do you guys know what the codon is for starting translation?
Methionine,
Yeah,
UAC, methionine.
it's AUG. Yeah, yeah, AUG, methionine. That's correct. Great. Uh, And during translation, there is an E site, right? So you have E, P, and U. So the E site in the ribosome is where what takes place with respect to the polypeptide chain.
Exit.
Very good. That's correct. So once you have added the respective amino acid with the help of the tRNA and it has formed its peptide bond, then it can move out from the E site and then the next one can come. So I think this is the last question, I think. Well, is this true or false? Transcription and translation can take place at the same time in bacteria.
True.
Yeah. Anyone has any contradicting answers?
It's a prokaryote.
That's correct. Yeah. Prokaryote. So I'm just saying in bacteria, but of course in archaea as well, that would apply. So that's exactly why, because it's a prokaryote. So all of the things that are important for transcription and translation are all just there in one big mess in the cytoplasm. And so naturally, transcription and translation can take place at the same time. And in fact, RNA pol has a docking mechanism that sort of allows this to happen at the same time. Unlike in eukaryotes, where of course, because these are segregated into organelles, these uh, different processes take place in different regions of the cell, one in the nucleus, the other in the cytoplasm. So that is the last question, I think. And this is just a little diagram of eukaryotic versus prokaryotic translation. Um, yeah, I think that's it. Yeah, that's all the questions.
Ma'am,
I thought I'd leave some time for you guys to ask questions if you wanted.
ma'am, can you repeat the answer of the 11th question?
11th question. Okay, sure. Yeah, so the 11th question is phosphorylation of the CTD or the carboxy terminal domain of the RNA polymerase. This allows promoter escape and elongation to occur during the process of transcription.
Okay, ma'am.
Yeah. So this all these slides and everything will be available, I believe, for all of you to access later on. I think they're shared with you, as well as these videos, like the tutorial videos are also going to be uploaded to YouTube, and I think those links will be shared with you as well. So if there's anything you feel that was left out or anything you want to go back to, those will be there. And right now, actually, I'm like the content I have prepared for all of you is sort of done. So if you guys have any other questions or anything you're confused about in the coursework till now, just let me know. I can answer that for you. And if you want to discuss any assignment questions, I think I'm not supposed to like tell you the answers, but I can guide you in the right direction. So if there's anything you want to know about the assignment questions, then let me know. Everyone's all good? Then I think we can just adjourn the session for today. Uh, yeah. So I will be taking these problem solving sessions on Mondays from 6 to 8 p.m. I think there are other TAs for this course as well who will be taking on different days. So please do join these. They'll be really useful for you in going through questions, going through the coursework. And next time I will prepare slightly more so that I can cover 2 hours. Um, but yeah, so let me know if there's any questions. You can also message. I think there is a Google group where you can put up your questions and doubts, and we will all be there to help you through them. I think that's all I had to tell you guys. So I think that's it then. We can end the meeting now, right?
Thank you, ma'am. Thank you, ma'am.
All right. Yeah,
Thank you, ma'am.
no problem. I hope you learned a lot.
Ma'am, if you have
Ma'am,
little bit time, I
Sorry.
came lately. I joined lately. I came
Okay.
lately.
Yeah.
So if the information is will be provided in the online in the Google,
Yeah,
I
so
might we
this
get the information.
information, yeah, so I think as a part of NPTEL, this recording, I've taken a recording of this entire thing. So this will be uploaded to YouTube, and I think the links will be shared with you. As well as these slides, they're going to be available as sort of Google links. You'll have Google links of some sort to um, a folder. So I think you will be able to access that. If not, then you know my name, so you can find my YouTube channel and I'll be uploading them there. So if you want to check it out there, feel free to do that. Yeah,
Okay, ma'am. Thank you.
no problem.
What about the assignment? I am the first. It's for the first time for me.
Yeah, so you're going to get one assignment every week based on the coursework that's uploaded, right? So you have
Uh,
your assignment inside the NPTEL portal, where the coursework is. So you have to fill that in. I think there's, I don't know, like 10, 15 questions. And I think the TA sort of sessions, these problem solving sessions, will help you kind of think critically and help you answer those questions. If you have more doubts that you want to get clarified, You can post your questions onto the Google group. I think a link was shared for that as well with you when you registered for the course. And you can ask us stuff there and we'll help you out, help you figure
Okay,
out the answers. Yeah.
okay, ma'am. Thank you. Thank you.
All right. Yeah. I hope no one else has any other questions, right? Then I will end the meeting here.